Hello everyone. So today we are in session 8. In the last session we talked about the endosperm. Few of you have a doubt regarding to the structure of endosperm. Why the micropilar and it will get change its position. It won't change its position, only for the diagram sake I just changed the location. Okay, it won't change the position at all. So, in the endosperm, the three types of developments I explained yesterday. One is nuclear endospermic development, and second one is cellular endospermic development, and third one is helobial endospermic development, and the helobial spelling was HELOBIAL, helobial endospermic development. So, in all those three, the structure and the description, just learn it everything. If they ask in the examination, just write it. And after that, after the endosperm, we are going to study about the embryo. Let me draw the diagram first. Again I am going to draw the embryo sac. Embryo sac will be, where it will be? It will be in the anatropous ovule. The embryo sac is now fertilized with the two male gametes, the egg cell and then polar nuclei. It got fused with the male gametes. The polar nuclei and the male gamete is done yesterday. Now today we are going to study the zygote. Zygote is the resultant product of the egg cell and male gamete of pollen grain. Here I will take this end as micropilar end, and this is the chalazal end. I will repeat once again, only for the diagram sake I am taking this position at the top and chalazal end in the at this point. Okay, in the ovule, in the flower, the embryo sac won't change its position. Towards the micropilar end, zygote is present, which is 2n in number, a set of chromosome it has. And then here yesterday we saw the endospermic development, how the endosperm develop. Only yesterday we studied how this development took place, how the endosperm developed, that only we observed. By the time when the endosperm develops, what the zygote do? Will it remain as it is, or it changes like the endosperm, how it develops? Likewise the zygote also develop. So here is the answer for that. Few angiosperm zygote, it will remain for a period. When the endosperm, when it develop completely by taking the nourishment from the endosperm, later on the zygote develops in angiosperm, in some angiosperm. But in some angiosperm, how the endosperm develops, meantime the zygote also keep on develops. Okay, so this is the single celled zygote. Only I'll take the zygote structure here. Here I am going to draw the zygote. I am leaving this embryo sac, and so this is the zygote. It takes mitotic division. So this is the one cell and one more cell is there. Now after this, let me write like that. Okay, we have structure here. So this is the stage wise development of embryo, where we can see the first thing, or at the first stage it was only one cell, and the next it has took place one division only, and then this don't forget we are talking about, or we are in the this part is known as micropilar, and this is the chalazal end part, and here also it remains same, and we can see a number of cells over here, and a little bit some flask shaped bulged portion is there at this point or at this region. This cell, this cell, it took place number of division, like mitotic division. So this portion is known as suspensor. 4 to 5 mitotic division or 2 to 3 mitotic division it takes, so that the cells divide. And so this is also one cell or basal cell, we can call it as. The shape what we are having now, that shape is known as globular shaped embryo. Here it was only one celled embryo or one celled zygote, here it become two celled. Now this cell took so many divisions, and this divisioned cells are known as suspensor, and bulged flask shaped structure is known as basal cell, and the shape is considered as globular shaped embryo. And then after the globular shape, the embryo again it takes division. How? So this is how it takes divisions. So this is the heart shaped embryo. So this is the heart shaped embryo. So we are having 3 shaped cells here. 3 shaped means this is not a different type. One after the other, the embryo has taken place, or it has attained a different structure. After the heart shape, it attains maturity. How? So consider we are talking about the dicot embryo. After the heart shape, how the dicot embryo, a matured embryo, looks alike. So that I'll discuss with you after some time. I'll tell you. After some time I'll draw the diagram of dicot embryo and then monocot embryo, so that you can see the difference between those two. So the thing is, you have to learn these stages first. And the premature, we can call it as a very young embryo, or the embryo which has not taken place any division, that embryo is also known as proembryo or proembryonic stage. And the stages or the formation of zygote, formation of embryo, the process is known as embryogeny. The formation of embryo is known as embryogeny. Okay, so we'll see the structure of dicotyledonous embryo now. So we are having 2 diagrams here on the board. One is dicot embryo and one more is monocot embryo. So this is a mature dicot embryo, and then one more diagram is there. So this is monocot embryo, right? So we'll study the structure, the parts of dicot embryo first. So this is known as the mature embryo. After the heart-shaped embryo, we are having a mature embryo here. So listen to me. Observe the parts over here. This structure and this one. These two are the cotyledons. It means these two are the two cotyledons and one embryonal axis. So this is the embryonal axis. And so cotyledons, after the cotyledons, what we are having now at the terminal position, we are having a plumule, and then it is followed by an epicotyle. So this is the epicotyle, and it is having one cylindrical shaped structure. Followed by the epicotyle, followed by a cylindrical shaped structure known as hypocotyle. After that, after the hypocotyle, at the terminal position, a radical is present. So this radical, also known as root tip, and the plumule is also known as shoot tip. This radical is protected by a root cap. It means, I think you come to the point, the plumule and radical, these are the points where the root tip and shoot tip arises. So these are all the tissues I should have to draw here. So no need to worry. If you are unable to draw the tissues, no problem. If you give a shade like that, that is more than enough. So that is the structure of a mature dicot embryo. And then coming to the monocot embryo, it is having only one cotyledon, and that is scutellum. One cotyledon it has, and an embryonal axis. So the scutellum followed by the coleoptile, you can see this coleoptile, which is a sheath protected to the shoot apex. So this is the shoot apex which get protected by the coleoptile, and it is also having an undifferentiated sheath, and the shoot apex is followed by the radical. The radical is nothing but root apex or root tip. And then what is that? So after the radical, it gets protected by a root cap. These two, the radical and then root cap, they both get undifferentiatedly covered by coleorhiza. It means it is having a sheath. The radical and root cap, they both get protected by a sheath known as, or it is covered by a sheath known as coleorhiza. So these are all the structures and parts of the monocot embryo and then dicot embryo. Write the diagram, follow the diagram, and draw the labelings, write the labelings. After this, I'll dictate some questions. That questions belongs to session 6, 7, 8, I guess. Okay, only 6 questions are there. I'll dictate now. Refer the sessions video, 6, 7, 8 sessions video, and answer the questions. The first question is, what is double fertilization? Describe fertilized embryo sac with a neat labeled diagram. What is double fertilization? So that is the first question, and it is having one more question with that. Describe fertilized embryo sac with a neat labeled diagram. You have to draw the fertilized embryo sac. It means in between the embryo sac, at the central position of the embryo sac, you should not write the polar nuclei. You have to draw, or you have to label the triple fusion, that is PEN. You have to show that, because in the question they have asked describe fertilized embryo sac, and then what is that, embryo egg cell, it get fused with a male gamete, that also you have to show, you have to describe that by writing one diagram, and it carries 5 mark. The question asked in the year 2014. And the next question is, papaya plants exhibit xenogamy only. Why? The next question is, papaya plants exhibit xenogamy only. Why? Xenogamy means the transfer of pollen grains from anther to stigma of one plant anther to another plant anther and another plant stigma of same species. It means xenogamy is purely cross-pollinated type, where in where the papaya plant, they exhibit or they are present in the nature. Different plants are there, male plant, one male plant and another female plant, they both when they pollinate, when the male flower pollen grains come and sit on the stigma of the female plant, female papaya plant, the cross pollination achieves. That all the things you have to write it in your answer, and the question carry 1 mark. And the next question is, third question is, what are cleistogamous flowers? Write their significance. Cleistogamous flowers, write their significance. Cleistogamous flowers, it comes in the autogamy, right? Viola, Camelina and then oxalis, these three are the plants name what I gave you in the example when I was explaining the autogamy. In that, cleistogamous flower is one type of flower where the flowers, they do not open, they will be in a closed manner, where these flower, they achieve self pollination. Okay, and the significance, just write it, write in your own sentences. We'll see how you will write. Next, the fourth question is, what is artificial hybridization? By which technique it is achieved? Artificial hybridization, by means of artificial, the pollen grains dusted on the stigma of the female flower or the flower by some plant breeders. This technique is known as artificial hybridization. And write their significance. By means of artificial hybridization, we can achieve or we can get a better characters plant, a high yield or better character plants we can achieve or we can get from this artificial hybridization. And the next question is, what is emasculation? And don't stick on to my answer, search it or refer the books. If you get a better answer, you can write the same, but you have to answer those. I am just giving you an idea so that you can write. Next question is, what is emasculation? Emasculation is nothing but, I told you in the artificial hybridization there are two techniques. One is emasculation and then bagging. In the emasculation, the anthers of flower, when the flower is in a bud condition, the anthers, it has been removed by using a sterile forceps so that we can control the self pollination. So this is how you just write it. What is emasculation? The emasculation question is for only 1 mark it is. And what is hybridization, artificial hybridization, by which technique it is achieved? I told you, no, that is 4th question. That question carries 3 marks. And then the last question is, write the characteristic features of insect pollinated flowers. So the characteristic features of insect pollinated flowers, the flowers will be highly rich in nectar, fragranted, or it is having fragrance, and it is colorful. So with these we'll end this session. Follow the diagram. If you have any confusion, ask me. Only a little bit I am doing so that you won't be worry. So because these are all the new concepts for you, it's little bit difficult to catch, so that I'm going taking very little concepts, or very small concepts. After this session we'll continue with the fruit and seed. Thank you.